Yep, they look great.
Yes. Perfect. Okay. Just remind that this content is covered by the copyright CC BY SA 4.0. So please read those terms if you're going to use this material. Welcome to Module 4. We're going to talk about antimicrobial resistance gene analysis. So this is really another type of annotation. You just saw a glimpse of it in Dr. Pico's presentation, where he was mapping AMR genes to a phylogenetic tree. So our learning objectives, we're going to understand the molecular basis of AMR. We're going to understand how reference databases are created and used. And then both at the web and the command line, we're going to predict resistance genes in genome assemblies from cultured isolates, but also resistance genes from highly fragmented metagenomic data like wastewater. And then if I can ask Charlie, just shout at me if I missed a hand up or a question on Slack.
No problem, will do. I'll try not to shout though, Andrew.
And then, I really want to highlight the promise, but also the pitfalls of phenotype prediction. My goal here, I want you guys to come away knowing what you can say confidently when you do these types of analyses. So we're not really talking about anything new in this regard. The theme is that bacteria are evolving resistance to drugs faster than we can bring new drugs to market. And so, tuberculosis, gonorrhea, right? These are things that are coming back. I've got a screenshot from former Twitter from Spain of a UTI infection, and you can see from phenotypic testing, with the except of ceftazide activactam, they were out of options, right? Everything was resistant, very few drugs. And that drug combination, for those of you in Canada, is not approved for use in here yet. You can only get it under special request. And they did one molecular test, the PCR for OXA48, which is a gene that confers resistance to a large number of beta-lactams. So I'm going to show some Canadian data. This is from a pre-pandemic study by the Canadian Council of Academies. So in 2018 in Canada, 25% of resistance or infections were resistant to the first drug attempted to treat. So by 2050, this is predicted that 40% that the first line drug that is, standard practice will not work. This was lead to about 400,000 lives, 120 billion in hospital costs, and 400 billion in GDP. This also doesn't highlight that what this affects on medicine. So you can't do a knee transplant or a lung transplant, chemotherapy, standard surgery if you can't control secondary infection. So this is all of modern medicine under threat. And these numbers are now out of date. During COVID, when you have a patient often dying of both a COVID with a secondary lung infection, you really had no choice. You had to use a lot of antibiotics to drive down the secondary infection so you could treat the viral symptoms. And as a result, we bred a lot of superbugs. And the reason that AMR is so difficult is the sheer diversity of chemistry involved. So we've discovered a lot of interesting chemistry to use as antibiotics, as antimicrobials, either to slow the growth of bacteria or to kill them outright. But resistance has evolved to all of these. So we're not talking about one mechanism. So while we're losing drugs through the resistance, this discovery pipeline to discover new compounds and new chemistries has been drying up. So we really haven't found something truly fundamental in scaffolds since the 80s. And the number of companies that are actually investing in antibiotic discovery is less than 5 on a large company scale. There's lots of startups struggling to do this. And from the discovery side, we go into often natural products, soil and water, and what we do is we rediscover the antibiotics we already knew. And medicinal chemistry has not, for the large part, not paid off. So while resistance is getting worse, no new drugs are coming along to replace the drugs that we used to. And as I said, this is a post-antibiotic era. So if we look at like my father and grandfather's generation, gonorrhea is not something worried about. If they caught gonorrhea, they had a shot of penicillin, right? And they were treated. So this is a bug that we have had a relationship with for millennia, and now making a massive comeback, right? So Cipro, we gave up on in 2007, Faxamine in 2012, and so on. And in fact, in 2000, I would get my year, 2012, I believe it was, There was a reported completely resistant strain in a high school in England, 14 cases, no drugs to treat. It is now a global a pathogen. It's in my community where you have completely untreatable gonorrhea. For a drug a generation ago, or for an infection a generation ago, we didn't even worry about. We just thought it was ultimately treatable. All of this is made difficult because AMR is a One Health and wicked problem. So One Health is, this is the Canadian confusogram, all the places that we have infectious agents, all the places we use antimicrobials, and how the genes and pathogens flow. So most of us tend to focus on the yellow circle on the bottom right there, the human, hospitalized community travelers. Another large fraction of us will look at the other sort of bad color vision, but looks orangest circle for food animals. So we use way more antibiotics in animals than we do in people. But these are connected through wastewater, through abattoirs, but a lot of people don't realize we use antibiotics in biofuel production, right? We have used it too often in swimming and aquaculture. And so it's a wicked problem because it's a multi-sectorial problem. There isn't just one place you can spend your money to combat AMR and reduce rates and lead to better outcomes. We really need to understand the transmission products of how the pathogens and these resistance genes flow through this complicated ecosystem. Compared to the British model, this is very simple. Theirs is even more complex, but I will say this thought, though, transmission models around AMR could be traced back to a paper by Linton in 1977, who had a rough drawing of this in his paper, drawn by hand, I presume, back in those days. So this is multi-sectoral. In Canada, we've made a commitment to look at genomic surveillance across all of these sectors to try and understand what are the drivers for transmission of AMR genes, in particular to identify hotspots where intervention would make a big difference. So that could be abattoir, that could be biofuel, that could be hospital. From the bioinformatics side, when we're doing genomics, the metaphor is actually fairly easy. So if it's through DNA sequencing, resistance genes can no longer hide. It gets hard in the details. But the technology we use is DNA sequencing. You've heard a lot about the different approaches, whether it be short read Illumina sequencing or long read nanopore. So it's important to pay attention to that market of how those technologies change, because that will change your capacity. But once you have sequenced, you're comparing it to reference sequences, known resistance genes and mutations. And that leads to the middle box where we can predict resistome. And this is the tool of antimicrobial resistance surveillance. And then the holy grail is the bottom. If we see those genes, can we actually predict phenotype, what drug to use in the clinic or at the farm? Can we use it for clinical decision making? But also at sort of like a national and international level, can we use that for risk assessment to track and see that certain trends are going on, that say we're losing macrolides at a disproportionate rate, and what is the driver behind that? From the science, we're really talking about three methods. Biocuration, which is the art of taking knowledge and turning it into algorithm and database, and that's what my lab focuses on. Bioinformatics, the ability to take a new sample, whether it be a swab or wastewater or cultured isolate, and predict what's in there. And then analytics to predict the behavior or the risk outcome. I'm going to review these three themes. I'm going to introduce a lot of concepts. We're going to follow up on them in the lab using both website and command line. So we'll come at these concepts a couple times. My lab produces the comprehensive antibiotic resistance database. With it, we have the software, the resistance gene identifier, and then we use machine learning for the analytics. So my group works in all this area. This is a Canadian course, so we're going to focus on Canadian tool. The CARD database is the most highly used, most highly cited, used about every 4 minutes. The superbug's going through it. Hopefully, for those working in AMR, you've had a chance to look at it, but I'm going to introduce you today in case you haven't. So the big thing to recognize, and this has been hinted upon in the talks, is we're dealing with a lot of mechanisms and drivers. So on the left of this figure from Jerry Wright are all the targets. Antibiotics target the cell wall, DNA, RNA synthesis, folate synthesis, the cell membrane, protein synthesis, you name it. There's lots of different targets in the cell. And we're always looking for antibiotic molecules that will hit new targets, right? Ones that we have no existing resistance. But on the right-hand side is what bacteria do. They've been using small molecules to talk to and to threaten and kill each other for millennia. So they've developed particularly efflux, just spit the drug out so the effective dose is too low. They can bypass it. They can alter the target so that the antibiotic can no longer bind. They can chew it up in pieces. And so you have a large number of mechanisms, some of which are very potent and some of them are very subtle. And that makes interpretation difficult. How resistance happens is simply misuse and overuse. So if we misuse, so that's as common, finish your course of antibiotics for 14 days. If we don't use antibiotics properly, there will be survivors, resistant survivors. We'll breed them. If we overuse, it's just simply an odds game. The more we use the antibiotic, the more likely a mutation will come along that's favorable for survival. And then as Rob has highlighted, what makes it a lot worse is these resistance elements, genes come associated with mobile elements. So if we look at a gene like NDM1, which takes out one of our two last drugs of last resort that we like to keep on the shelf, it was discovered in a single patient, a tourist in India who then traveled to England for additional treatment. That one case, N of 1, is now known from 30 some odd pathogens through mobile genetic elements. So here's NDM1. Again, appeared in a single Swedish tourist, and we're not sure of its source. Had initial treatment in India, flew to England for more. It was in Klebsiella. This is our surveillance work, all the pathogens that it's known in. And if you notice, say beside A. baumannii here, there's G's, P's, and whole genome shotgun. So it's known in the genome. In other words, it's incorporated into the chromosome. It's known on plasmids, and it's known in shotgun sequencing. And notice how many of these have plasmids to it. No genomic islands yet for NDM. So, and as I said, when you have an infection with this drug, you're down to one drug class of last resort. That's colistin. You really don't want to use that drug. It's really rough on the kidney. So you hope you don't get an infection with this gene. In my neighborhood alone, a city of about 600,000, we have over a dozen plasmids carrying this gene, rotating, flying around in clinical settings. Okay, some of you may not know this gentleman, but the older ones will. So I'm going to talk about this. Was the Secretary of Defense since George W. Bush's era, talking about the Iraq War. And he did this quote, and the media sort of just laughed at him that he was avoiding to answering the question. So he said that when asked about issues, what's going to happen in Iraq, he said, well, there are no knowns. There are things that we know we know. There are no unknowns. That is to say, there are things that we know we don't know. And then there are the unknown unknowns. There are things we don't know we don't know. They thought he was avoiding the question, but really he was citing black swan theory, which is a way at looking threat of how you assess threat. And this metaphor works really well for surveillance of AMR. So if we look at the known knowns, these are the genes and pathogens that we routinely track. Now, my local public health diagnostic lab, they do culture-based detection for about 8 different pathogens. They do PCR detection for around 14 different genes. These are the most prominent and prevalent cases that cause problems in the hospital. That's a very tiny number. So the known from an epidemiological perspective is quite low. The known unknowns are the genes we don't routinely track. So as you're going to see, CARD tracks thousands of genes and variants. So we know these are often involved infections. We know they're out in the community, but we're not tracking them at any level. And I don't really need to explain post-COVID, there's always variants. So you may start with one resistance gene, but quickly there will be variants and novel mutations. And because often we're doing PCR, not sequencing, this is all flying under the radar. And then unknown unknowns are emergent threats. So in the 15 years I've been working in this field, we really had two. The NDMs, which took out one of our classes of important beta-lactams, drugs of last resort, and then another gene called MCR1 that took out our only remaining drug of last resort, which is colistin. These are unknown unknowns because often they're involving completely new mechanisms or unexpected impact to it. And we often don't know where they're coming from: soil, water, clinical overuse, you name it. So that, there's a bit of mystery. And you'll point out we need to get better. NDM1 is called the New Delhi metal beta-lactamase because that's where it was originally reported. That does not mean it originated there. Geoshaming is something we need to get rid of in this field. But this is where we're at now, and sequencing promises to open up all of this. So when you sequence, say, a cultured isolate, we will detect those same targets that we do with PCR or culture in a clinic, but we also will detect all the variants that we track sort of in our database. And then the big question is, how can we build bioinformatics to predict emergent threats, things that are flying under the radar? There's a real promise to this, and we saw, we had a sort of a 10, 15-year plan of clinical use of these data. Then the COVID pandemic hit, and now every neighborhood hospital has a DNA sequencer. So this has really changed. This is now that we're doing this. And the reason we've got this course running because there's a training gap, right? There's a lot of information. And so I'm going to walk through to make you comfortable in sequencing in this setting. All right, following up on some of the earlier lectures, I'm going to give you sort of three axes that you can look at analyzing the resistome. So the resistome means the complement of resistance in genes and mutations in your sample. The first is from perfect matches all the way to novel emergence. So perfect matches, in other words, it matches identically amino acid to amino acid, an enzyme that's been fully characterized and published. In the middle, you have novel functional variants. It's a variant of a known enzyme, and then really novel emergent. In public health, we really focus on the left side, right? Tracking ones that we actually can point to science and that we understand exactly what they're going to do and what the clinical outcomes. In academia, we focus on the right side, right? Trying to understand the incoming threat that's out in the environment. The next axis is whether you're looking at acquired resistance genes that are often on plasmids or mobile genetic elements. These are usually highly expressed and usually have large impact in animals and people. And so a lot of the PCR we're doing are often plasmid-borne beta-lactamases, for example. In the middle, there's resistance by mutation, which is generally on the genome. So pathogens like TB or Neisseria gonorrhea generally evolve resistance by point mutation. And so we often we have a sequencing or at least sort of like a LAMP assay to detect the individual mutation. So again, it depends a bit on the pathogen. The last is more intrinsic resistance or changes in regulatory response and efflux, spitting the gene out. This is actually some of the hardest to predict. A lot of surveillance work ignores these. We might know intrinsically certain drugs won't work in Pseudomonas, take that as a given, but we really don't understand how Pseudomonas, why it upregulates efflux for certain drugs and not other drugs, and exactly what genes are involved. The last axis is technical. Are we doing a culture-based whole genome sequencing? Do we get really high quality assembly data, whether it be de novo assembly or guided assembly, but we get really high quality genomes of a cultured isolate that's causing the problem that we're seeing. So we get whole AMR genes and alleles. This is really the easier end of the bioinformatics. Are we doing whole community sequencing on the right, which is metagenomics? So now we're sequencing a swab in the neonatal ICU, or we're sequencing a soil sample from a farm or wastewater from a municipality. This tends to be short read, tens of millions of sequences on a Illumina platform. So now you don't have complete genes or genomes, you have short sequencing reads. So where's the field at? So all on the right, I'm putting in green. We're all, not just CARD, but all the NCBI resource, ResFinder in Denmark, all our colleagues, all the specialist database. We're doing really well in this space. So culture-based after plasmid-borne or highly expressed high-quality threats with perfect matching to say, oh, I've got NDM1 in my isolate. This is working extremely well. It's very effective for the Enterobacteriaceae and the escape pathogens. Which is it covers a huge range of the resistance we see clinically in animals. Where we're doing reasonably well and a lot of improvement in the last year is mutation-based, TB in particular. Between the Cryptic study and the World Health Organization synopsis, we actually now host the global data set of TB. I'm going to show that to you. We have really excellent criteria for evaluating mutations in TB and knowing that if you see this mutation, what's the likelihood that you lose specific drugs. Neisseria gonorrhea, not as much. We know those mutations, but trying to make a good predictions on the outcome, we need to make improvements. On the right in red is where we're really this is the sort of the future. This is graduate work. So AI and machine learning showing a lot of progress to if we do surveillance in our communities based on DNA sequencing or surveillance on farms or wastewater, we're getting closer to get early warning prediction of emergent threats. We also, through some brilliant experimental work, like the Cox Lab at Guelph, for example, or the Pittock Lab in England, really breaking knowledge on how efflux works and how the regulatory, particularly pathogens like Pseudomonas that infect, say, a CF patient, for example. So we've made good progress, but there's a lot more to be done. So you should be a little more skeptical in any bioinformatics that works in these areas. I used to keep metagenomics in the red box as well, but that's really changed in the last 3 years. We're doing quite well. The bioinformatics, the reference data for working with metagenomic data, particularly in wastewater, but that's having lots of spillover to studies in One Health, so by waterways or farm or what have you, is doing really well. We're getting cheaper sampling methods, better bioinformatics. Where we are stuck are things like sepsis. So sepsis, to give you the general statistic, after 4 hours of sepsis, your chance of dying doubles every hour. You don't have a lot of time. There are well over 100 pathogens associated with sepsis. It only takes a small amount. It's not like they're overwhelming your blood. We are stuck on doing metagenomics on sepsis simply because getting the material, the sequencing, and doing the bioinformatics can't be done fast enough yet and cheaply enough to beat that timeline. So this is one of the holy grails. And this is not just a bioinformatics challenge. This is going to be collaborative with sample processing and sequencing technologies to break the back of sepsis. Okay. We talked, there's 3 areas. There's biocuration, bioinformatics, analytics. I'm going to give you some examples of each. And again, we'll do a bit of this in the lab. So let's talk about this. Many people come into the field. They're thinking, well, I need to analyze the AMR. They do a little lit search or online, and they find an overwhelming amount of databases. So my lab has been working with Wikipedia to update that list. There are many. Many are boutique. In other words, they're for very specific problems, like the BLAT database is excellent for engineered beta-lactamases. There are excellent tools for TB, for example. Um, there are ones that are, they pass. They're active and curated for a year or two, and then they lose their funding and they're not curated anymore. Some of them are synthesis, so they're databases built of databases. But the big three are in the National Library of Medicine's NCBI Pathogen Detection Reference Gene Catalog. That's in Washington, D.C. And ResFinder in Copenhagen, Denmark, and the Comprehensive Antibiotic Resistance Database in Canada. These three all work to be comprehensive. ResFinder really started with plasmid-borne, but they've really branched out and are doing all types of mutation. These three databases collaborate with each other. They exchange data with each other. We trade standards. We often collaborate collectively on that. And so you can do well by any one of these. I'm going to show you mine, but it's not that my colleagues in Denmark and the US aren't extraordinarily excellent as well. There's a lot of effort in the community to harmonize and standardize. We all have slightly different areas of evidence. NCBI focuses on supporting the US ecosystem, the USDA, EPA, Defense. ResFinder has a European-centric. We work on a Canadian global. We tend to work on a commercial side. So CARD. We're going to look at this, but this is a screenshot of CARD's database. The idea is it is a resource of the mutations and genes underlying AMR. Generally, up until recently, the entire focus has been bacterial drug resistance. We've now collaborated with the Fung AMR group. So this is the first sort of coalition of mutations underlying fungal resistance. We're hosting their data. We're slowly incorporating it into CARD completely. And then we've worked with the International Consortium to synthesize all the tuberculosis data. To get a gene or mutation in CARD, there must be a peer-reviewed publication in PubMed that we can point to and cite, and that publication must have clear experimental evidence of elevated MIC, in other words, increased resistance relative to a control. The caveat there for those that are more clinically bent, that doesn't mean that elevated resistance is going to cause a critical problem, right? That's a difference. That's a question of breakpoints. At what level is it considered resistant from a dosage perspective? Many of the genes in CARD are just that. The resistance we curate is well beyond breakpoint, but we also track all the more subtle resistance that get poured in the literature because we expect it to evolve to get worse. For those who work on beta lactamases, the genomic era has hit this hard. So because of genome sequencing, we discover beta lactamase sequences in their hundreds per day. The NCBI leads the international effort to characterize these. So a large portion of the beta lactamases genes in CARD and others, you can't point to a paper or an experiment to it simply because of the pace of genomics. And of course, the sequence has to be available in GenBank. And I always love showing this screenshot. This is 7 days of CARD usage by public health agencies that I snapshot by doing it. It's a really excellent community. We have online forums that we share with NCBI and ResFinder that involve academics, companies, public health, where we work together to keep up with the data. So if you're in the AMR space, please help us contribute. I will hide these stats. So CARD has 6,442 reference sequences. So these are the canonical alleles that, when they express a protein, cause resistance that are described in the scientific literature that we understand mechanism and resistance. But our surveillance effort, when we analyze all the available genomes after, actually is just shy of 300,000 alleles. So there are a great many sequences out in farm and clinic and the rest that are floating around that are variants. And those variants are presumed to cause resistance, but there's no experiment behind them. We spend a lot of time trying to put confidence around that. But it is important for you to consider when you find a resistance allele in your sample, is it one that you can track to an experiment and paper through CARD or ResFinder, or is it one that is a variant that you should be a little suspicious that it might not behave the same way phenotypically? Okay, so from the data science point, CARD has a knowledge base. This is Brian, the lead curator. So we use ontologies. You've heard a little bit about ontologies and data standardization. So this is the ANT6 aminoglycoside resistance gene family. This terminology always has a unique ascension, has a definition and any synonyms. And then the knowledge base tells you if there's what genes are within it, in which case there are 4, but also what family, what resistance, what drug classes, what mechanisms that go on. This computational sort of graph of knowledge allows us to do really interesting bioinformatics and to analyze your samples, for example. So this is a data harmonization and standard effort. So if you go to public health agencies in Canada, when they're recording data on prevalence, they're using the terminology in CARD to track things. We work with GenEPO and IRIDA and lots of efforts to harmonize data. If we look at one family to it, what's guaranteed in CARD that any gene or mutation will be classified by its AMR gene family, its resistance mechanism, its drug class. There's almost none that are not curated drug class. Where the curation is less complete is the relationship between individual genes and mutations and individual antibiotics. We're about 4,000. I should update that slide. But essentially, you're talking that our human curators would have to read over 13,000 papers a year just to keep up with that. No surprise, we're starting to work with natural language processing to do that. So it is a gap in the field all the databases are struggling with, which is to keep up to date on the actual phenotypic reports for individual genes and mutations. And of course, we have the sequence underlying all that. On top of that hand-curated data that connects to paper, we do in silico molecular epidemiology. So we're looking at the ANT6-1A enzyme in CARD. It's coded by a single gene. We do what's called CARD-R. We take just shy of 20, this is an older data set, 25,000 chromosomes, 2,600 genomic islands, 48,000 plasmids, and 172 shotgun assemblies for 414 pathogens from NCBI. And we analyze them all and we find 279,000 variants. But this now tells us which genes are in which pathogen and which variants are in which pathogen, which are associated with genomic islands or plasmids or chromosomes. And this is a baseline amount of epidemiological information. For those who use it, we are literally running our biannual update right now to update this data set. So what does CARD offer? High quality reference database on the molecular basis of AMR. It's expert curation. We trust is our business model. So other than that molecular epidemiology, which is done by algorithm, everything is hand curated. Every paper's read individually. We do have the CardShark text mining algorithms that make sure we don't miss any papers. But when CardShark says, "Hey, read this," it's a human that reads it. We cover all of AMR data and mechanisms. We include advanced analytics for AMR gene discovery. And outside of genomics, we provide tools for data harmonization or standardization for machine learning through the antibiotic resistance ontology. Dedicated curators, we update one to every 3 months, depending on how much is happening in the AMR space. Okay, bioinformatics. So going to highlight this case. We have sort of clinical genomics rounds, and this case was out of this traveler that had managed to get typhus in the spine. And turns out, they'd had a complicated travel history, had misused their antibiotics, weren't entirely honest all the time. And this was a bizarre infection with really bad outcomes. So being good bioinformaticians, we asked for it to be cultured. We sequenced this salmonella that was in the spine. There's the genome there. And we ran it through our resistance gene identifier software. Now, what RGI does is takes this genomic sequence and compares it to the comprehensive antibiotic resistance database, looking for genes and mutations. And this is what we got back. So the box is on the outside of this visualization. We're going to do some of these today. Each one is a resistance gene. And if it's in green, it's a perfect match to a published enzyme, a complete amino acid match. If it's in yellow, it's a variant of that. So compared to your Albage salmonella, this is not normal. This is a superbug, right? This is a scary perspective. But you actually can redesign these data. So now the middle circle is the drug class. And on the outside, it says how many genes are undermining that drug class. So if you look, like cephalosporins are being undermined by 7 separate genes, for example, penams by 12 genes. How did this bioinformatics happen? Well, we have that knowledge graph connecting genes to drugs in CARD. So it just reanalyzed the original circle, this one, from a drug class and drug perspective. So again, from a drug perspective, this is a superbug. There aren't a lot of drugs left to treat this patient. And I will say clinicians did succeed and fully treated it, and there was a full cure for this patient. So you saw that we have these perfect stricts. So let's go back to black swan theory. I'm a threat surveillance guy. So the perfect matches that are in green are our known knowns. They match known reference sequences in the database. The yellow is strict. So these are known unknowns. We know that variants are out there. We know some of these variants have never been sequenced or reported in CARD. So we call it strict because it's similar to the reference within a model. We think it's a functional variant. A lot of science, a lot of biocuration try to decide where those cutoffs are. I'm not going to go into that too deeply. And then outside of that, there are loose hits, which we didn't show for this isolate. These are the unknowns, things that have only a degree of similarity to resistance genes. Most people never look at the loose. The only time they look at the loose is when the perfect and strict cannot explain what's going on with their isolate or their patient. And then they think they might have something new. Let me look in the loose to see if there's anything. But most of it's junk. Most of it's just weak sequence similarity. What's unique about CARD compared to some of the others is we curate data in an algorithmic perspective. So this is the ANT61A. This is an enzyme encoded by a single gene. So either you have this gene or you don't in your pathogen's genome. So really the data for this model is simply the sequence, the DNA and the protein sequence, and what we call a blast cutoff, a bit score cutoff when we compare the sequence to the reference in CARD using the BLAST algorithm, which is the standard sequence similarity algorithm. We curate that carefully, that cutoff. So if the BLAST score is below that cutoff, it's not similar enough to be a functional variant. Maybe it's a proto-resistance gene that someday we'll need to worry about, but it's above that cutoff. We predicted that it's a functional variant. So we have to code that. There's a manuscript just about to come out in preprints talking about evaluation, whether this is a good model or not for predicting functional. I will say we worked very hard at these. We really do suspect, and the preprint supports, that when we call something at strict, it overwhelmingly is strict, a functional variant. But if you want to be absolutely sure, do be aware, a strict hit has never been cloned and expressed and directly test. So if it's critically important, maybe that's what you should do. And if you'd go to the CARD website, we actually, with Jerry Wright, have a platform for cloning and testing individual alleles that we're happy to share. Now that's a resistance by acquisition of a gene. Maybe a plasmid brought it in. Other resistance, in this case, like a gyrase B are part of the machinery of the cell. They are targeted by antibiotics. What they do is they change their amino acid sequence so they're still functional. They can still do the job of gyrase B, but the antibiotic can no longer bind. So these are resistance by mutation. So again, we do a blast-based homology detection with a bit score cutoff to find the gyrase A, but that's not enough to report it. Once we find it, we compare it to a catalog of resistance mutations, changes in amino acids, and sometimes combinations, right? So we have this codependent signal resistant variant. So you need more than one mutation. So in the case of gyrase B mutations, if you want, say, to cause resistance to fluoroquinolones, the dominant one is this one at the top, the E at position 463 turning to K. That's the dominant resistant, but these other co-variants. So all this data is hand curated from the literature to be, and so that's under a what we call protein variant model. So all the data in CARD is we curate everything, but you need to keep up on the software side on RGI. We break everything down by models. There are 8 fundamental model types. I'm going to highlight, though, even though the data is in CARD, only 4 of those model types have software in the RGI software to support them. So the bottom 4, knockout, non-functional insertion, gene clusters, and efflux pump systems, RGI is not predicting those. It might predict the individual genes underneath of them, but not everything. And even the top 4, protein homolog, protein variant, RNA variant, some of them have asterisks, so not all mutation types are being annotated, such as frame shifts. We're literally very close to taking those asterisks off and predicting all possible mutations that cause resistance to those genes. So there is a link between mechanism resistance and the genetic encoding, and therefore the bioinformatics you have to use. Okay, so we just looked at a cultured isolate. Let's look at something like culture-free. So I got a picture of a swab there that I use in the neonatal ICU or a municipal outflow, right? So in this case, when you sequence a complex sample, the vast majority of the material is background DNA or host DNA. So when I sequence a swab from a baby in the NICU, guess what? Most of the DNA is the baby. Do that, but a large fraction is also the bacterial microbiome that belongs inside a baby, bifidobacterium, etc. Same thing in wastewater outflow. There's background bacteria that have nothing to do with disease. So the pathogen slice and the AMR slice of that is very tiny. Finding AMR genes in these complex samples, needles in the haystack. The solution in the past is to sequence a lot. So if you sequence tons, you're raising the detection level to find those rare DNA molecules. Therefore, it can be very expensive. The DNA is very fragmentary, so this is shotgun sequencing. So you get 250 to 350 base pair fragments, so you're not getting whole genes. Makes the bioinformatics harder. Also, I'm going to show you that the analysis is sensitive to the reference data. So the NICU, if you are, that works really well because you're taking clinical AMR that's probably in a baby, and CARD is generally clinical AMR references. It works well. But if you take a forest soil sample, the AMR alleles that are sitting in a forest sample or in a lake in the Arctic are very different from what might be in CARD's reference data set. Then a corollary is great. We found an AMR gene in these samples, but we actually need to know which bacteria were carrying it, right? And remember, we only have fragments, little pieces. It's hard to make policy decisions if you don't know which pathogens actually involved in the fact that you're finding AMR genes. So the question of where did that 250 base pair gene coming from is something we use called the Burroughs-Wheeler transform that came out of human genetics. In other words, you've heard the term read mapping. Common software is the Bowtie 2 families, the BWA families. Essentially, it's an algorithm that uses basically as a quick computational method. I'm not going to get into the math, that would be cruel. But it aligns that short 250 base pair to a reference genome. So originally it was designed for doing exome sequencing in humans for cancer. We have a reference human genome, we shotgun sequence their tumor, and we align all those short 250 base pair reads to the human genome, and we look for mutations from that. It's computationally intensive, but can be very accurate. But in AMR, it doesn't work well. So if you're doing anything in metagenomics, wastewater, I want you aware of this, what's called the allele network problem. So this is a screenshot of the TEM beta-lactamases. This is a major family conferring resistance to beta-lactam antibiotics. And there are hundreds of variants, and each one gets its unique name. This is, again, an old slide. I think it's over 400 now. And the key thing, these variants evolve from each other. They have slightly different resistance profiles, occasionally dramatically different resistance profiles, which could come down to a single mutation difference. So they are 99.9% identical. It's one or two or three mutations that differ to them. So they are a network of alleles. So the Burroughs-Wheeler transform wasn't really designed with that in mind. It was designed to a human genome where the only duplicates it really needed to worry about were like alpha tubulin or elongation factor 1 alpha. They was not designed with an allele network in mind. So we went ahead and examined this. So this is Amos Raffenia. He actually simulated metagenomic sequencing of 7 AMR genes using shotgun sequencing at 10-fold coverage. So these 7 genes, he simulated 10 sequencing reads of 250 base pairs each. And then he used bowtie 2, the standard Burroughs-Wheeler transform, to align them to card. And notice the 7 he simulated have the red arrows, but what CARD's RGI gave out was a huge range of AMR genes. And then on the y-axis, there's a thing called the MAP-Q. It's a quality of performance, and a MAP-Q of 35 is quite low. What we've discovered is that when you use bowtie 2, so say we're looking at the simulated CTXM15 gene. That algorithm figures out it's dealing with a CTXM for sure, that family, but then it gets lost in that allele network. And a lot of the reads, it aligns to the CTXM gene, but sometimes it guesses and aligns one to the CTMX101, the CTMX114, the CTMX157, CTMX3, etc., making it look like there's a bigger diversity of AMR genes in the sample than there actually are. So if you're reading the literature using metagenomics and bowtie 2 or anything like that to assess AMR in a soil sample or water, be skeptical of their gene list, because until recently, people were not paying attention to the allele network problem. So who paid attention? Our colleagues in Denmark, extraordinarily programmers, the ResFinder team, came up with a new algorithm specifically for AMR, but really any allele network problem, called KMA. So what KMA does is the Burroughs-Wheeler transform to get it in the ballpark, and then it uses k-mers, which Gary von Domselaar introduced, to be more precise. So this is the exact simulated data. We simulated 7 genes, and the annotation from RGI, when it uses KMA to align those sequencing leads to card, got 7 genes. I will say 2 of them, it got the wrong variant. It got the most closely related variant, but it was still off. It's not perfect. But now the MAP-Q score is 194. Our estimates for abundance is much better. And so this is as close as we get. We're not overestimating the number of genes in a metagenomic sample because of this improvement of paying attention to the allele network. It is now the default in our software. But I do want to point out those greens. Sometimes the difference is so subtle that it still can't quite figure it out. Right. And so it wasn't cat I we simulated, it was a different cat we simulated. So that was part 1 of the allele network problem. There's part 2 of the allele network problem. Canonical card, what we curate in the card by reading the literature, by expert curation, doesn't represent the real diversity of AMR. The literature is overwhelmingly clinical, human beings in a clinical setting and their isolates being sequenced. Therefore, as a reference set, it's not that great for doing a farm. It's not that great for doing, a wildlife or doing river systems or Arctic ecosystems. It's essentially good for clinical and wastewater, because wastewater really reflects clinical. So this is why Brian did the card resistomes. This is why we use a huge amount of compute to get hundreds of thousands of genomes from repositories like NCBI, many of which are non-clinical, and we annotate them to find all the variants. As I said, there's about 279,000. Many of these variants now belong from non-clinical isolates that are more interesting. So you can use this as a reference set to be more representative. If you don't, you will under-curate your data if you're not in a clinical setting. So if you're doing metagenomics, this is our suggestion. You do your sequencing platform to get your millions of metagenomic reads. You can use the card canonical if you're strictly worried about clinical types of data, clinical isolates. But if you're not, you add the card in silico variants. The card are from all the isolates, not just clinical. So you can choose what your reference is. This gives you the read counts against the reference sequence. So your data could look something like this. Out of my millions of reads, there were 42,000 aligning to the TET-Q, 77, just over 7,000 to TET-X, for example. This is the standard type of data you see from metagenomics, and we're going to look at some today. The key thing is these algorithms, even the KMA, are not designed for SNP detection. So they really only work when it comes to dedicated resistance genes like an enzyme that encoded. You're not going to get your gyrase B mutations, right? There's a lot of us groups working on to add SNP detection, but be warned. When you do metagenomics with KMA aligners, these methods, you will analyze the non-mutational type of AMR only. So you're not getting a total resistome prediction. So if you're working in tuberculosis, this is not going to help you at all because TB mutation is overwhelmingly, TB resistance overwhelming mutation. This preprint came out from Mateus wearing his medal. He was a varsity athlete. That once you find these genes, but what pathogen? So is it E. coli? Is it Acinetobacter? Is it Pseudomonas? Using all those variants, those hundreds of thousands of variants, Mateus and an undergraduate built a KMER tool looking for subsignals saying, oh, this variant of TM1 is only found in E. coli. This variant of TM1 is only found in Pseudomonas, and done a prediction of pathogen or plasmid tool, so pathogen of origin prediction. So this tool is out. It's finally validated. The preprint's out. It's going into peer review very shortly. This allows you to do metagenomics to not only catalog the AMR genes, but to make some likelihood predictions of which pathogen is carrying those AMR genes because your data is so fragmentary. One of the ones we're interested in is if we get enough data that you sequence enough, if whether we could do sub-assemblies to actually assemble the complete AMR allele sequences. If we have complete alleles, then we can do mutation work, right? This is some interesting bioinformatics. The reason I got it in blue, it actually depends on lots of sequencing data. So, it's not fair to ask people to spend 10,000 per sequencing run of metagenomics. So we need a breakthrough here on the sequencing cost to get depth of data so we can start to do allele level assemblies of metagenomic data to do SNP detection, for example. Before we get to that, I'm going to highlight that this is computationally intensive work. So our website, CARD, doesn't do metagenomic analysis. I don't have enough chips. But we partnered with the Chan Zuckerberg Foundation. So as long as you're not in industry, there is a website with a lot of compute behind it, does microbial genomics, including metagenomics of resistance using CARD and RGI. It is free, it's confidential, it's got lots of good security. It's designed through the nonprofit models to support public health and research surveillance of AMR and other things in metagenomics. So just highlighting that out, that if computation is a limit, this is a really good solution. It will do normal RGI as well for cultured isolates, example, and a lot of the tools that you see in faculty demo in this course are on this pipeline. Okay. Last little bit on metagenomics. We talked about it's expensive, right? I want better AMR surveillance data. I need it to work out risk and to decide how government should spend their money. I need metagenomics data at farms and wastewater and rivers and trees, et cetera. So I got to make it cheaper. So we collaborated to make a bait capture method. So bait capture is making RNA fishhooks. You have your metagenomics library full of millions of molecules, most of which are not AMR. We used CARD to design molecular fishhooks that would only bind to the AMR alleles. So when you do an in silico or in solution hybridization and you wash it out, everything that is not AMR washes out of the tube, but every AMR DNA sequence is kept in the tube by these molecular fishhooks. So when you sequence, the sample's now enriched or concentrated for the AMR target, which means you have to sequence way less more to get your signal, makes the metagenomics cheaper. So this is the first paper led by Alison Gitor. We did some clinical shotgun. So the 6 columns on the right are standard shotgun sequencing. Every row is an AMR gene, and you could see the brightness of the blue is what we detect. We had a hard time detecting a lot of the AMR. We used the molecular fishhooks to concentrate the AMR samples in the AMR molecules in the sample, did the exact same amount of sequencing, and we had huge enrichment. Our detection levels went up. So we are now to practice for metagenomic and AMR. Instead of sequencing 10 million molecules to get detection, we sequence roughly 100,000. We've cut the cost at this level down a tenfold in that regard. Now, there is the version up two. The other part that made this as expensive was synthesizing the molecular fishhooks. That's generally done by commercial companies. And so that costs a lot. We actually invented a new in-house synthesis method that's available. So overall, we've reduced the cost of high-resolution metagenomic sequencing of AMR by 2 orders of magnitude. It's a lot cheaper. Plus, we build a software pipeline that every time CARD updates, it will update the probe set that you can use. This is all available on the CARD website. So if you're in metagenomics, we have the bioinformatics. We just went through it with KMA to analyze the data, but we've also worked with lab people to make it just simply more higher resolution, better detection, and fundamentally cheaper to do so. Okay, last little bit, checking my time. We're going to talk about analytics a bit. So this is Dr. Kara Zhang, and we asked a simple question. If you analyzed a bunch of E. coli genomes, 115 from our clinics, and we ran them through RGI and it told us what genes are mutations, RGI then connects that to individual drugs. If we just took it as given that CARD is always right, if you see the gene, you lose the drug, what actually happened when you culture them on a plate and test them is just a simple rules-based approach saying if you see the gene, you lose the drug. Does it work? So this is for 18 drugs. If it's gold or yellow, we were wrong. So we predicted it was resistant and it wasn't, or we predicted it was susceptible and it was resistant. So you could see we underperformed quite badly. We really didn't do really well on any drugs, ertapenem maybe a little bit. What that shows you that the connection between genotype and phenotype is not simple. When you're doing resistome surveillance, you're getting the resistance list of genes and mutations. It's a long way from being able to know how that bacteria will behave in an infection or even on a plate. That has to do with transcription and expression of it, has to do with efflux and other things. So rule number one, you're getting an annotated list when it says this gene's connected to that drug. That's a possibility. But when we trained it using a machine learning approach, so machine learning looked for patterns that we didn't see through in CARD and those resistance genes and reweighted them essentially, and then analyzed more samples, we actually got very well. So give me an E. coli sample from Hamilton, Ontario region. If I sequence this genome and run it through CARD in this machine learning, I have a very high likelihood of predicting exactly which drug will pass and fail in that setting. What was interesting about the machine learning is it predicted things we didn't know. So CTMX15, from the literature, we knew it created resistance to 3 drugs. The machine learning predicted a 4th one, cefazoline. And in fact, there were 5 other beta-lactamases where the machine learning predicted a broader spectrum of impact. We collaborated with the right lab who has the antibiotic resistance platform. So this is an expression platform and an efflux deficient E. coli. And lo and behold, the machine learning was right. We were all already frightened of CTMX15. It's an important gene we tracked. And this paper showed through machine learning that was worse than we thought. It had a broader range of activities. But I do want to highlight this analytics. There's a lot of literature you can read, and it'll appear that it's all over a place. So we repeated this with Pseudomonas aeruginosa, a bacteria that particularly we see infecting CF patients. Repeated the study, and I'm not even going to show you the graphs. Even with machine learning, we couldn't get to good phenotypic prediction, which basically means we're measuring the wrong thing. There's areas of biology that we just don't know. It's not necessarily genomic that's controlling it. Maybe it's epigenomics or biofilming or things like that. In fact, what we've learned that even the E. coli that looks really good, if we try to run that machine on a different municipality, it doesn't work well because the genetic background is different for E. coli in that community. So in a practical sense, if you think it won't work for all pathogens, even if it works for E. coli, you'd have to retrain the model for every municipality. But AMR changes probably every few years. You'd have to rebuild it all in every municipality because AMR evolves. That's financially not feasible. We got a long way to go for the analytics side. Okay, last thoughts. So if you're going to do this work, context is everything, right? So this is why it's great that Rob did that lecture. Plasmid-borne AMR genes are generally highly expressed and highly impactful. So if we see them plasmid-borne, they tend to be the high-threat genes. But you got to understand behavior. Just because you're full of resistance genes, if you biofilm, like Pseudomonas, those antibiotics can't get in anyway, right? And then you have decision. If you get a list of 50 resistance genes you're isolated, how do you deploy that knowledge? How do you decide what to follow up on? Technology important. What's the volume and breadth of your culture capacity? Can you do it anaerobic, for example? What's your computational? Can you do read mapping and genome assembly, right? Where you have to make some designs on what lab work you're going to do based on what computational work you're capable. And how do you keep up with 5,000 plus AMR genes? We at least try to help you with our database. Confidence can be a real challenge, right? So when you're doing SNP detection, say in TB metagenomics, sequencing error is a thing you have to take into account, right? So we saw that early on in Jared's lecture of aligning reads to figure out variants. Plasmid genes are very easy to find, but we've heard already that resolving plasmids genomes is very difficult. We now use Plasmidosaurus. So this is, we send a DNA isolate. It does Illumina and nanopore simultaneously. 9 times out of 10, we get complete plasmid sequences. So this is going away as a problem. Lastly, you be very careful on the genotype phenotype. A lot of SNPs are potent on their own, but some are additive. You will always find efflux genes. Some are important, some of them are not, some of them are additive. How good is the underlying knowledge? A resistance gene that only has one published paper, yeah, be skeptical versus one that's been involved in many studies. So the genome and plasmid at the first pass, right? So nanopore sequencing is cheap and fast and is awesome. It requires GPU processors, and those are getting more expensive all the time because of the AI bubble, we'll call it. You combine it with Illumina, you tend to get really good complete chromosomes and plasmids. And we've heard already about MobSuite and others that we're getting better tools to understand plasmids. Culture can be a limit, right? You have many limited culture capacity. So bait capture is what we just showed to increase effectiveness. So we're starting to bypass culture and only do bait capture. Particular things that are a pain to culture or a pain to isolate, the H. pylori or C. difficile, we're going to bait capture or nanopore-based methods. But if culture is involved, like how do you handle a big outbreak? I will say that metagenomics is a big part of our collective future, whether it be environmental metagenomics or swab, unbiased or bait. What we are seeing that sequencing costs are going down. That market's in our favor. Though I would say, Illumina library construction costs have not dropped significantly in a while. And the algorithms for read alignment are improving. Pardon me. And lastly, I did a lot of pandemic surveillance work and really struck home to me that there's an inverse relationship between speed and scale. So while sequencing prices are dropping, library prep costs are not.